So it's a pleasure for me to present the two speakers of today who kind of worked together at some point and presenting different aspects of their work. And the first speaker would be Michael Heizinger. Michael is a researcher in Germany, so he obtained his master's degree in bioinformatics from the Technical University of Munich. And he completed his PhD in the same university under the supervision of Professor Burkhard Rost, whom I know quite well from many, many years ago. Michael's work dealt with applying large language models to protein sequences, employing transfer learning techniques and approaches to using protein embedding from protein language models. After he finished his PhD at the end of 2022, Michael continued as a postdoctoral fellow in the Rost lab, and he worked on various problems at the intersection between biology and AI. Since the beginning of this year, Michael took a big step. He has been working as a machine learning researcher at Sanofi Biologics in Frankfurt. So Michael will present his work today on what he called PROST T5, these kind of bilingual language models for protein sequence and structure, which leverages the protein language models to simultaneously model both 1D sequence information and 3D structure information. So Michael, the floor is yours.
Hi, perfect. Thanks a lot for the kind introduction and for having us. Let me just quickly bring it up. So you see now my screen?
Yes.
Perfect. Okay. Without any further words, I will continue right away. So I will talk about the work that I've been doing since my defense. So essentially over the last year, I've been working on how to apply the language models that we've been developing over the last few years in the Rost lab, also to protein structures. Okay. Perfect. So, but maybe just a brief historic background to also put this work into perspective. So this was the status quo when I started my PhD around 2018. So here you see the development of three big databases that are relevant for protein science. So the development of the size of the data points in them over the years from 2011 to 2018. So in blue, you see the amount of structures that we have. So experimentally resolved 3D structures, which is it's appearing like 0, but it's actually 100K. But looking at the scale of the y-axis, you see why it's appearing like 0. And this is actually a feature. It's not a bug. So also scaling this logarithmically would have worked, but the effect is intended. Because what we want to show here is the large gap between the available number of sequences that are actually part of the UniPark database and the lack of information that we have for those sequences. Because after all, we only know essentially that there is a sequence, but we have no further information about the structure or the function of these sequences. So the idea of how we can bridge this gap was essentially over the years always very similar. So we try to essentially predict this sequence annotation gap. So this is also known as the sequence structure gap, but on a more general level, you can also just call it sequence annotation gap, because for most of these sequences, we have no information except for the sequence. So how this was usually handled was that you have an input protein sequence, you somehow encode it, make it machine readable, you feed it to a machine learning device here depicted as a feed-forward neural network with an input layer, two hidden layers, and an output layer, and out comes some sort of prediction that is of interest to you. And just for visual reasons, I put here the structure, but this could be also function. This is what people have been working a lot on over the last years. But the question that I asked during my PhD was rather, how can we actually represent proteins? So less about the central part, so which machine learning device to put here, whether it's a random forest, a super vector machine, feed-forward neural network, but rather, how do we actually go from these amino acid sequences, these tokens, to something that is machine readable, that a computer can understand, because computers usually do not like these letters, they work with numbers. And people have been trying various approaches over the last years. Most famous are all these approaches that rely on evolutionary information, where you gather evolutionary related protein sequences in large databases, and then you accumulate the information in these multiple sequence alignments using statistics. So for example, position-specific scoring matrices, or a bit more complicated evolutionary coupling scores. But after all, this is not what we want to go for, because after all, I started working on protein-protein interaction prediction, and many of these approaches do not really work well if it comes to handling paired input. So we wondered, are there any alternatives on how can we actually make protein sequences machine readable? And around the same time also, there was a big revolution in natural language processing, where all these language models came up. So for us, this has proven to be extremely useful. So we jumped on this train and we took techniques that were developed for processing human written text, so natural language, and adapted it to work with protein sequences. Because after all, from a purely machine learning perspective speaking, both are just strings of tokens, so discrete input. And we made the best experience with a model that is called T5. It is a big transformer consisting of an encoder part and a decoder part. And from a high level, it works very similar to BERT, which might be a bit more famous. So you essentially corrupt, or you give the protein sequence as an input to your transformer. You corrupt certain amino acids in the input, or words in the case of natural language processing. You feed this corrupted sequence, where you have just replaced a single amino acid by some special token that hides the identity of this amino acid at this position. You feed this corrupted input through the transformer, and the task that you optimize for is that you ask the model to reconstruct which amino acid was at this specific position that you've corrupted, given the uncorrupted context. So you always keep a certain fraction of the input correct, so you do not modify it, but you only modify certain fraction of the input. And then you train this model on reconstructing these corrupted outputs. And this you do for millions or even billions of examples. So we trained this on a large set of metagenomic sequences, and we fine-tuned it then on UniRef50. And there are also certain clever techniques which are for T5 working better than for BERT, so the relative position encoding. But on a high level, it's essentially just a cool way of making use of these large protein sequence databases, because the big advantage here is during this so-called pre-training of the protein language model, you do not need any further information except for the protein sequence in the input. So as you are essentially just training on the sequential signal of the input itself, you do not need any further information on the 3D structure of the protein or its function or anything. You can really just train based on the sequence. And for sequences, we have, as you saw on the previous slide, hundreds of millions or even billions if you go to the metagenomic space. And now the question is, how do we actually make use of this? And here the step 2 comes in, so the so-called feature extraction. So once you've trained this model PROT-T5 on billions of protein sequences and reconstructing these, you can use it for a so-called feature extraction. How this works is that during inference, you feed a protein sequence through your language model. And for us, this was only the encoder part of PROT-T5. So we removed the decoder part, and then you extract the hidden representations from the last layer's hidden state, which we call embedding. And this is essentially just a numerical representation of the proteins, of the protein input sequence. So just from a high-level perspective, for each amino acid in the input protein sequence, you get a numerical vector of a certain size. And for us, the size was 1024. And this is something that nicely feeds into a feed or in a neural network, for example, or any other prediction device. And we've showcased that this works well with secondary structure and subcellular localization, but by now we have it also for many other prediction tasks. And only recently we've also explored so-called parameter efficient fine-tuning, where you also backpropagate through the language model. But this is only a side tour. What I actually want to come to, and this is just the background section, because after all, I'm building on these language models. While we worked on building many predictors on these language models, something happened, which as most of you might know. So AlphaFold 2 happened, and this pretty much closed this sequence structure gap that I had been referring to previously. So after AlphaFold had been released, and also after the AlphaFold database had been released, we had predicted structures for more than 200 million protein sequences, and this closed the gap between the large number of unlabeled sequences and the relatively small number of where we had experimental resolution 3D structures. And one of the main motivations why we started working with these language models in the first place was actually that we saw that we had a unique chance of leveraging the gigantic amount of unlabeled protein sequence data that we had available. And suddenly, when the AlphaFold database was released, we saw that there might be a second chance for something similar, because after all, we suddenly had a second modality that describes these proteins available on this large scale. And the question was, how can we leverage this information properly? So how can we now make actually protein structures machine readable? So not sequences, I don't want to say we solved, but we essentially had now a tool available that allowed us to make sequences machine readable. And now we wondered how we could actually do something similar for structures. But the question is, these transformers, they work well with tokens. So they need discrete input. They were developed for processing natural language, which consists of words which can be tokenized. And this is also why it was easy to adapt it to protein sequences. However, protein 3D structures are much more complicated. So even though we can essentially describe a protein as a one-dimensional string, in reality, a protein lives in a cell actually as a 3D entity, and only by forming this 3D shape, it can also perform its function. However, this is something that is not easily parsable by the existing transformers. So of course, by now we have also these more complicated versions thereof, like the SE3 transformer, which can process 3D data, but this comes with other problems, like you can also not have an all atom input, for example. So we wondered, is there any alternative? And by coincidence, Martin Steinegger developed shortly before this a tool that's called FoldSeq. So some of you might also know this. And this is by now, at least to my knowledge, the fastest structure comparison tool. And what made it very, very interesting for our goal of essentially tokenizing 3D structure was that it also uses internally a machine learning device called vector quantized variational autoencoder, which converts 3D information to 1D. And then it applies essentially the MM6 pipeline. This is also what makes it extremely fast. So how does this work? So you loop over all the residues in the protein and over the residues in the protein structure. And from there, you find the neighboring residues using a so-called virtual center, which is based on the side chain. So sort of the gravity center of the side chain. And from there, you extract something like 10 features, which give you the distance of the residue that you're currently looking at to the C alpha coordinates of the neighboring residues. It also gives you the distance and the angles thereof. And this you feed into a variational autoencoder. A variational autoencoder is essentially just taking input feature space, it compresses it to a smaller space. So you can imagine this here in the input, you have like 1024 inputs, just for the sake of the example, it's not 1024 here. Then you compress it and then you try to reconstruct it. What makes this special is this discretization. So this is also what makes it a vector quantized variational autoencoder, because you enforce that in here, in this bottleneck dimension, you have discrete tokens. And after training this on the PDB, you can apply this to arbitrary protein 3D structures. You loop over each residue, you extract these angles and distances, you give it to the encoder, and then what you get is actually in the bottleneck dimension, this discretization essentially of all the residue 3D coordinates. And here on the right, you see, because Martin cleverly chose this bottleneck dimension to be 20, so you map essentially an arbitrary input to 20 bottleneck dimensions. And on the right here, you see the symbolic representation of these bottleneck dimensions. So, and they coincide with the amino acid alphabet. And here you see that some of these tokens align really nicely. Some others might be referring more to a loopy structure. But after all, you essentially now have a second alphabet, which coincides to have the same letters as the amino acid alphabet, but now describing not the amino acid identity of a residue, but rather describing its 3D shape, its local 3D shape. And with this, we were perfectly equipped to essentially map, or we were using also predicted structures so that you could go from the 20 amino acid, the tokens via AlphaFold to a 3D structure. And then we use FoldSeq to go back to a 1D string. Now consisting again of these 20 3DI tokens, not of the 20 amino acids. So you can imagine that for each protein, we now have essentially two strings. One string describing the amino acids, the other string describing the local 3D shape of each residue in this protein sequence, which we derived from AlphaFold predictions. So the first step towards training now a language model on this new data modality was to create a data set. And for this, we were building up on the existing clustering of the AlphaFold database that was also provided by Martin and his colleagues, for which they used the AlphaFold database with 214 million protein structures. And they used MMSeqs to sequence cluster them, or cluster them based on sequence similarity, which led to around 50 million cluster representatives, for which they then chose always the protein with the highest PLDDT as a representative. And the resulting representatives were further clustered with FoldSeq at 90% structural overlap, or an e-value below 0.01. And this led to essentially 19 million cluster representatives, of which 16 million were singletons, so only a single protein structure was in this cluster, and around 3 million non-singletons, where you had more than one protein per cluster. Given that we considered this, we considered this as an extremely good starting point for any machine learning project, because it boiled down the 214 million to a representative and very diverse set of 19 million proteins. So this way you could save some compute, because you do not necessarily need to iterate over the nearly identical proteins that might be part of this. And you could, it's just cheaper. Computationally, it's cheaper. But still, we wanted to have something more than 19 million, which is why we expanded all the clusters by the 20 most diverse members. And this is possible with HHBlitz, for example. After we did this, we performed some further quality filtering. So we removed any protein from our data set that had a PLDDT below 70, that had a length below 30, and that had a very repetitive 3DI token string. So some 3DI strings, if you are describing an all alpha helical protein, are always just having the same token. And this we consider not very informative for our goal of training a transformer on top of this, because otherwise this would be very similar to giving a string to a language model that is always consisting of the same token in the input. After doing this filtering, we ended up with 70 million sequence structure pairs. So we had 17 million amino acid sequences and 17 million corresponding 3DI sequences. For the sake of, because the alphabet was identical, and in order to make it easier for the model to actually learn that there is a difference between these two entities of tokens, one describing amino acids, the other one describing the local 3D environment, we casted the 3DI tokens to lowercase for the sake of training. So how did we now actually train or fine-tune the model? So we started from the existing Prot5 model that I have mentioned previously, which is a large transformer consisting of an encoder and a decoder. And we continued the task of mask language modeling, or in the case of T5, it's a bit different. It's called span-based denoising. But after all, the idea is always the same. You corrupt tokens from the input, you give the corrupted input to the model, and you ask it to reconstruct the corrupted tokens. And this we now did not only for the amino acids as it was done for the protein amino acid sequences as it was done before, but we also did it for the 3DI strings. So it was essentially two tasks, but the same model. So we always used the same Pro-T5 model, and we just essentially created batches that had both amino acid sequences and 3DI sequences. And the third step was then to actually translate from one to the other. Because here, what you learn here is just to compress the information of the 3DI string or the amino acid string by itself. But you never tell the model actually which amino acid string goes with which 3DI string. But after all, we have this pairing, and you can imagine this as two different languages. One language speaking protein sequence amino acids, the other one speaking protein structure 3DI. So we decided to use this setup as a sort of, or we try to mimic the translation approach that is also quite famous in natural language processing, and also T5 is essentially built for this, because you can give the model in the encoder input one language and ask it in the decoder output to reconstruct the other language. So this is depicted here by the coloring. So for example, you give it the green input 3DI string, and in the output, you ask it to reconstruct the green amino acid string. And vice versa, you can do the same for the other direction. So you can go from the amino acid string to the 3DI string. In order to make it a bit easier for the model to learn which direction we are currently interested in, we also introduced two prefixes, one telling it to go from amino acids to 3DI and one telling it to go from 3DI to amino acids. But it's the same model, so it's always a single model that was optimized on two tasks simultaneously. So now you might wonder, what do we do now with this? So we can now essentially get these 3DI strings from the structures, we can give them to the model, but now, so what? First use case is in line with what we've been using language models also so far, so we can use it for feature extraction. But the interesting aspect now is that we can extract features not only from amino acid sequences, but also from 3DI sequences. So how we did this is we actually, very similar to how we used language models so far, is we gave the model, and for us, it's again just the encoder part of Pro-T5, or now we call it actually Pros-T5 because it's a protein sequence structure T5. So it should be still resembling the original name of Prot-T5, but also giving it the structure spin. So this is now Pros-T5, and it's just the encoder thereof. So we dropped the decoder completely. You can give the Pros-T5 encoder now either an amino acid sequence or a 3DI sequence. You feed it through the encoder, and you again extract these embedding representations, these vector representations from the last hidden state of the encoder, and use this as an input to a prediction device. We still use secondary structure, even though that this is a completely boring task by now, given that AlphaFold can give you 3D. But for us, it was still a useful benchmark, because we saw from previous experiments that the better you do in secondary structure, the better you usually also do in other tasks. And secondary structure prediction is just very well established. We have good data sets for this, and it's very clear how to work with. So we just decided to still look at the performance thereof. So here along the y-axis, you see the Q3, so the three-state secondary structure prediction accuracy. Along the x-axis, you see three different language models that just takes protein sequence as an input. So this is currently not showing you any 3DI information. It's just ANC and ESM2 and Pro-T5. Which are just three different state-of-the-art protein language models that can turn amino acid input into vector representations. The error bars here are given by the differential performance on three different data sets. So the lowest performance is always CASP14 performance. The point here in the middle is always CASP12 performance. The uppermost bar is a data set that we called new 364 that we introduced in ProTrans. But this just, so these three data sets should give you some sort of idea about the expected error depending on how similar your protein is to the training data. And you see already that these language models all perform rather similarly, I would say, with a slight improvement of ESM2 over the two T5-based models, ANC and ProT5. So now we have also ProT5. So ProT5, by just feeding amino acid input to the language model, getting these embeddings and training to predict secondary structure from it, nicely showed that we actually saw some minor improvement for predicting secondary structure using the embeddings of this model. But the interesting spin is now also that we can also encode 3D structure. And yes, this becomes very circular. I get to this in a second. But you can now essentially take also a 3D structure. You can use fold-seek to extract the 3DI string from it. And this 3DI string you give to ProT5, you run it through the encoder, you get the embeddings from the last hidden layer state, then you predict the secondary structure from it. And you see that we're approaching 90% Q3 with this approach. But you might also argue, okay, fair enough. You have 3D information, you give it to a model and you ask it to predict secondary structure. This is sort of boring or stupid, up to you. And this is why we also decided to just check this by just one hot encoding 3DI and training the same network on top of the one hot encoded 3DI and checking the three-state secondary structure accuracy. And you see that we already get very competitive with this extremely simple approach. So you might not actually need this fancy transformer over here. You can just get away with one hot encoding 3DI and you already get a good secondary structure presentation. We also did a slightly more in detail analysis of this using the Skanky plot, where we showed that these how many of these 3DI tokens are actually part of these three secondary structure states. So you see that V, for example, has a strong prevalence to be part of a helix, while the D here has a strong prevalence to be part of a loopy region or undefined other region. But still, we wanted to see whether we could also predict 3DI, because after all, we trained on the translation of amino acids to 3DI strings. So if we could bypass this problem, this information leakage problem, which requires AlphaFold structures to extract 3DI, if we could just by the same model itself predict 3DI, and we could feed this into the model and get the similar performance, then this would already be much more interesting again. Unfortunately, the predicted 3DI turned out to be a bit less informative on this one. You can still compensate for this by concatenating, because what you can always do is just you can take the amino acid ProT5 embeddings, concatenate them with the experimental 3DI embeddings and give it to the model. This is very, very strong predictor, but again, it's using essentially 3D information in the input, which is not clever. But you can also get quite far with just concatenating the amino acid embeddings of two different language models. And yeah. So so we always took the 3DI from the AlphaFold prediction. And the P3DI is always predicted by our model. But we can also do a bit more interesting things with this than predicting secondary structure. So for example, we can check how well we can predict in which CAF homologous superfamily. So CAF is this hierarchical classification of proteins or protein domains according to their secondary structure composition and their sequence similarity. And we tried previously to just use the distance in embedding space as a classifier. So you have a small set of labeled proteins and you have a set of unlabeled proteins, and you just embed both, and then you compute the Euclidean distance between the two sets, and then you transfer the annotation of the closest neighbor in the annotated set to your query set that are unlabeled. So very similar to homology-based inference, but just replacing the notion of sequence similarity as means of annotation transfer by embedding distance. And this is what we did here. So here you see the performance of actually for a test set, a non-redundant test set to a labeled set of lookup proteins for the homologous superfamily level of CAF. You see how far you get with just doing sequence similarity via MM-seqs with this. Here is again ANC, which is a T5-based model. ESM1B, which is the previous version of ESM2. We did not run the ESM2 for this one, unfortunately, yet. Here is our PROT5 that we proposed, and here you see the improvement for this task for our newly proposed PROT5 if you just use amino acid sequences in the input. And here you already see a nice improvement over the previous version, and this is also maybe a bit more interesting of a task than secondary structure prediction, because after all it boils down to remote homology detection without having to predict 3D structure in this point, because you just need to run a forward pass through this model, get the embeddings, and do the pairwise Euclidean distance between the two. Now you can also compute the Euclidean distance by feeding in the 3DI derived from the AlphaFold predictions. This is even much better than just using the amino acid input. But again, you need already 3D structure, which is expensive to produce. And in this case, the predicted 3DI and the embedding there of the Euclidean distance thereof also already provides a very strong classification accuracy for the homologous superfamily. You can also improve this further with contrastive learning. So you essentially just optimize the embedding space such that proteins that share the same homologous superfamily are clustered closer, while the others get pushed further away. But I do not want to go into the details on this one here. Unfortunately, we repeated the same exercise, but now not for something that is heavily structure related, such as secondary structure prediction or Kerr homologous superfamily classification, but more function related. For this, we used the binding residue prediction task that we had been also working on in the past. So we already had numbers from previous experiments where we already ran ESM2 and PROT5 and ANC. So these are all these language models that work only on amino acid sequences. And here we have then the performance of the proposed PROST, once running, getting embeddings for the amino acid sequence, once getting embeddings for the experiment or the AlphaFold predictions of the 3DI and the predicted 3DI. But in all cases, you see that we cannot reach the performance of the best performing model, which is PROT5 in this case. We can compensate for this partly by concatenating, but currently my status is that you win something if you have a strictly structure-related task, such as the classification of Kerr families or secondary structure, something like this. If you have a task like this, then the embeddings from PROST5, I would be confident that they will help you. But if you are rather aiming for a function-related task, then I'm not so sure whether it's worth the overhead. But after all, it's also relatively easy to derive the embeddings for the amino acid input. But this is only the usual use case, which is just involving running a forward pass through the transformer and getting embeddings of it. The more interesting part actually comes from this translation aspect that I've also mentioned, so that you can now actually go from the language of amino acid sequences to the language of 3DI structures, essentially, using this model. So you feed in an amino acid sequence and you get out the predicted 3DI string. And now we wondered what happened actually if we feed this predicted 3DI string to FoldSeq and use it for search. And we benchmarked this by replicating the FoldSeq benchmark, where they measured the sensitivity up to the first false positive, or the fraction of queries that reach the sensitivity for the SCOP40 benchmark. And here you see the performance for the family, the superfamily, and the fold level of the SCOP hierarchy. And you see three different methods. In yellowish, you see the sequence-based method MMSeqs. In black, you see the performance if we run FoldSeq on 3DI derived from experimental PDB structures. And in red, you see the performance of our proposed PROST5. So the cool thing is that we are getting very close to the performance, the remote homology detection performance of FoldSeq that uses experimental structures, but we're using our predictions instead. So this is pretty cool because you can bypass this expensive 3D prediction part. The problem is just that this approach is still relatively slow because of the sequential nature of the decoder. So while, and this is now very technical, but the encoder can essentially get embeddings by just a single forward pass through the network, while the decoder works more like LSTM, a long short-term memory network, which is always predicting the next token, then it's feeding in its own output, and then it does this until it reaches some output state. And this sequential decoding makes it rather slow, so it's still, compared to AlphaFold predictions, relatively fast. So it takes around 2.5 seconds for a protein of length 400. But this was still, we could do better. So what we decided, we went for the same approach that we did also previously. So we essentially just gave an amino acid sequence to the PROST5 encoder. We got embeddings, and from these embeddings, we were directly predicting the 3DI alphabet. The big advantage of using this iterative decoding is that you can actually also tune the diversity of the output. So this is very useful if you want to generate different diverse output. But what we are actually interested in this case is we just want to do remote homology detection. So we do not want to have a variety of outputs. We just want to have the 3DI string for this amino acid sequence. And this is perfectly fine if you then just use the encoder for this. And here now we've added one line in blue. We use the predicted 3DI from the CNN method. And you see that this is essentially falling between the black and the red line. So we can now do we can reach the homology detection sensitivity of full-seq that uses experimental structures, but we are using these predictions, and we can, and Milot showed this nicely, we can actually do this for whole proteomes in minutes instead of days that ColabFold would have needed. And I think for the sake of the time, and I'm so sorry, Milot actually also took a video of this where he showcased that this is now already implemented in the FoldSeq web server. So you can actually use this feature already now. And it's also part of the FoldSeq toolbase already. And this is extremely useful if you have large metagenomic sequence databases that you want to search with structural sensitivity for a protein of interest. So what you could do, for example, if you have an enzyme that is very interesting to you, you predict its structure either with ColabFold or you derive the 3DI string with PROST5, and then you search with PROST5 against large metagenomic sequence databases that would not have been possible before because you do not have AlphaFold predictions for these large metagenomic sequence databases. Okay, and for the sake of time, I will just very quickly go now over this. Is there a chat? Yeah, okay. Yeah, so you can also do inverse folding, so you can translate in the other direction. So you can go from 3DI to amino acid sequences, and this you can then benchmark using the self-consistency that was also already introduced by the protein design community. So you predict the 3D structure of a design sequence using ESM fold, you compare it to the starting point, and this gives you some notion about whether the generated sequence is actually useful or not. We did this also for our model. Turns out it works okay-ish, but actually the protein MPNN, which is probably some sort of state-of-the-art for this task at the moment from the David Baker lab, is definitely better for this. And with this, I just want to thank you for your attention. I want to thank the co-authors, Konstantin, Joa, Milot, Adrian, Burkhard, and Martin. And this is just a picture of the black smoker. This is actually where this organism that we run the proteome for lives. So it's from the deep sea.
Thank you very much. That was really very interesting. Okay. Now, thank you, Michael. So we are moving now to the second talk by Céline Marquet. And Céline earned her bachelor's degree in business and engineering from Karlsruhe Institute of Technology and a master's degree in data engineering analytics from the Technical University of Munich. And currently she's a PhD candidate in bioinformatics under the supervision of Dr. Burkhardt. And she's working on her thesis that, deals with scalable protein variants and phenotype predictions using language models and alignment-based deep learning. She recently finished an internship with Vant AI in New York City, which must have been really interesting, applying DL techniques and system biology methods. where she worked on latent. I mean, that's what I just said is what this company is doing. And at this company, Céline worked on a latent diffusion for conditional protein generation and protein-protein docking. But I don't think she's going to talk about that today. But, anyway, so generally her interests lie in enhancing speed and accessibility of in silico protein function assessment, and particularly the investigation of mutational landscape for disease and design. So today, Céline will talk to us about her work on protein embedding for the prediction of binding regions in disordered proteins. And these are disordered and structured proteins, and they are really, multifunctional often, and only, certain amino acids, participate in these interactions. So I saw this application early on, and this is what attracted me to hear about what she was doing. So Céline, the floor is yours.
Thank you so much. Can you see my screen?
I see a general screen. Yeah, this is your screen.
Okay.
Yeah,
okay,
good.
Wonderful. Thank you so much for the kind introduction. So as Shoshana's already said, I will talk about how we use protein embeddings to predict binding residues in disordered regions. And this was work done with Laura, and then Michael, who you've just heard from, and Burkhard was the main supervisor. And I'm very excited to talk actually at this webinar because I know Elixir is very involved in intrinsically disordered proteins. So I will keep this motivation relatively brief, but why are intrinsically disordered proteins so interesting? Well, first of all, they lack, at least in some regions, stable secondary or tertiary structure. And overall, it's a very complex and context-dependent phenomenon. So it doesn't mean that disorder is always the same type of disorder. So it can be kind of hybrid regions that change upon binding or other factors, and that makes it very challenging to determine even a ground truth. So that makes experiments very challenging, but also in silico prediction tasks, because if you struggle to determine a certain type of ground truth, that also means it's more difficult to benchmark methods. On the other hand, these proteins or protein regions often play key roles in molecular recognition and binding interaction. So they're very interesting for drug target development and other disease-related studies. And so here, for the sake of time, I'll do this briefly, but I just put an example of where you can see the disordered residues on the left in red, and then the binding residues on the right in purple. And this is a lipase example from a database called MobiDB, which I will also touch on again later. And yeah, so why do we look into in silico tools in this setting? So as in many cases, these tools are great complements for more time and resource intensive experimental analysis, and there are already a lot of community efforts going on that strive to standardize resources, make them more accessible. So on the left here, I took a plot from an overview from the Elixir intrinsically disordered protein community, where you can basically see different databases, different efforts to just centralize those within the community. And then on the right is a rather popular benchmarking effort called Critical Assessment of Protein Intrinsic Disorder Prediction, which is led by a group, the biocomputing group at the University of Padua, and it's a community-based blind test to determine the state of the art in predicting disordered regions and residues involved in binding. So far, there have been 2 rounds. The third is ongoing. So in the first round, 11 methods were evaluated for binding regions for 646 targets, out of which 414 had no binding residues though. So they had disordered annotations, but no binding annotations. And then in the second round, 15 methods were evaluated, and they had 348 targets overall. So those were for the disorder challenge. And then, unlike in the first iteration for the binding challenge, only 78 sequences were considered, and they all had binding residues. Doesn't mean all of the residues of each protein were binding, and it doesn't mean that all of them were disordered, but each of them had at least one residue. And then, so why do we want to use protein language models? It's not just for this use case, but overall, for many prediction tasks in the space, in silico tools rely on evolutionary information, which is often derived from multiple sequence alignments, and then homology-based annotation transfer. And this information is just not always available. So this isn't necessarily only related to disorder, but you can often encounter residues or entire proteins where you just lack alignments, or you lack similar proteins where you already have annotation that you can transfer to your problem. And this is the nice thing about the protein language models, which you've heard about from Michael, that you can basically take one sequence and always generate these protein embeddings for them, and you don't have to rely on multiple sequence alignments anymore. And we have used these language models now for a lot of use cases, one of which was predicting binding residues, which was work done by Maria Littman, who was also a PhD student in our group and has since graduated. And she used PLMs to overcome some of the challenges of traditional methods, which is not only that you might lack alignments, but it could also be that inference is really slow, and just generating the necessary input can be very challenging. So this is why protein language models have successfully been used in other spaces as well. But what Maria noticed with her method is that her binding predictor, bind embed, and also other binding predictors based on MSAs or other input, which were mainly developed on data from ordered regions, often fail for these disordered regions. So the goal of our project was then to develop a binding predictor specifically for disordered regions. And as usual, the main part of this work was generating or extracting a fitting data set. And luckily, one of the databases in the Elixir infrastructure is MobiDB, which aggregates disorder and binding annotations from literature, experimental evidence, and then also predictions for all known protein sequences. Which is amazing for us because we could extract curated disorder annotation from there, where we had a cutoff in October 2020, and there we had roughly 3,000 proteins. And at the time, the KITE-1 challenge, the results were out, and Anchor 2 was a very high performing method, so state of the art. So we decided to compare against this method and then also deep disbind. And we then chose their training sets, or we considered their training sets when we built our test set so that we could fairly compare against those two methods. So for the test set, we first randomly chose 200 positive and 200 negative examples from the data set. By positive, we defined any protein that had any residue that was annotated as binding. And negative means there was disorder annotation, but no binding annotation. And again, doesn't mean that all residues were disordered or binding, but just some parts of the proteins had to be. And then we performed 2 different ways of redundancy reduction. So one more rigorous way was a redundancy reduction based on sequence and structure. So there we used MMSeq2 and UniqueProt. MMSeq2 is an alignment-based method, and UniqueProt is a clustering method, which also takes structure into account. So what we then did was we did this redundancy reduction for the test set, so the positives and the negatives, but then we also redundancy reduced this against the training sets of deep disalign and anchor, and all excluded proteins were added to the train set. And then for the train set, we performed a redundancy reduction that was a little bit less rigorous. So we chose a redundancy reduction tool, which is probably very well known, but CD-HIT at 40% sequence identity. So this was only sequence-based, just so we wouldn't lose too much training data. And for the finalization, we decided to adjust the positive and negative ratio for the training and the test set, which left us with 150 negatives and 80 positives. And then for the train set, once we had finalized our test set, we redundancy reduced that again with the sequence and structure-based MMSeq2 and UniqueProt. So just to maybe go back, so the training set itself is just redundancy reduced based on sequence, but then the test set is redundancy reduced based on sequence and structure. And looking at the train set versus the test set, that again is then structure and sequence based. So overall in training, we had a bit more than 1,000 negatives and 697 positives. And now finally to the method development. So this is a setup which has worked well for us in different settings, also here. So what we do is we basically take an input sequence, which you can see on the left bottom. We feed this into a language model, in this case PROT-T5. And this language model is pre-trained, so we don't have to do anything with it. We don't use it for training. We just use it to generate our input, which is these embeddings. So those are just matrices of a shape, which is the length of the protein times 1024. And then this is input to our method called IDBindT5. And we add one more dimension, which is order and disorder annotation. So this is just a binary indicator whether a residue is in an ordered region or a disordered region. And our output is a binary per residue prediction score. So for this kind of setup, or as it usually is the case in machine learning, you kind of have to go with the data availability. So it was necessary to break down this problem based on the data that we have. So first of all, we saw that if we use mostly ordered proteins for training, we're not doing very well in disordered proteins. So this is why we had to generate an entirely new data set. And then instead of differentiating between certain binding partners, so whether a residue binds to a protein or nucleic acids, which some methods already do, we only had binary annotations. So this is why we also only get binary predictions out. And then another assumption that we made was that we know whether a residue is ordered or disordered. And obviously, this isn't always the case. What we have done is use predicted disorder as input. This also works, but this is then obviously dependent on how well your prediction tool works. The architecture is pretty simple. So it's just a feed-forward neural network, 3 layers. You have an input layer, you have a hidden layer, and then the output layer. So this is very straightforward. We tried other things, but as for other use cases, we've seen that these FNNs, very lean FNNs, work pretty well. So here you can see the performance on the test set. So in blue is our method ID bind T5, then orange is anchored to, green is deep disbind, and then this red one on the right. So this is a random baseline where we just shuffled the residues. And as you can see, ID bind does perform pretty well. The black bars are the 95% confidence intervals. And while it's numerically highest, as you can see, it's not statistically significantly better than deep disbind. It is often a little better than anchor 2, which I will show you on the next slide as a nice feature, because so we also assessed the speed of the methods. We took a subset of the human proteome of 11,000 proteins, which we filtered from MongoDB at 100% sequence identity. And then we took proteins which were shorter than 5,000 residues and had binding annotations. And we did this on our server, where deep disbind took more than 33 hours for this prediction. Anchor 2 took 21 minutes, and ID bind T5 took a bit more than 2 hours. So as you can see, while we're better in terms of performance on the test set than Anchor 2, and similar to deep disbind, we're actually much faster than deep disbind. So we do think this is a very nice feature of ID bind T5, especially for large scale applications. And then we also, we tested performance on just a consumer laptop, and there, I think it was 16 GB of RAM and 4 cores. Took roughly 2 seconds for a protein of 400 residues. So that's also nice that you can do this locally. And then by the time we had developed our methods and also worked on the preprint, everything, Kite 2 results came out. And here I just briefly wanted to show you performance on this data set. So Kite 2 only has a time cutoff. There is no redundancy reduction in terms of sequence or structure. But what happened is basically our cutoff from MongoDB was October 2020. Their cutoff, I think, was November 2020. So that worked out really well for us. So we can use this to compare. And the picture is very similar. So ID bind T5 and deep disbind perform similarly, while Anchor 2 is a little bit worse. And then there have also been other methods in Kite which outperformed Anchor 2 and deep disbind slightly, but because we'd already worked on this and the method performance were all relatively similar, we chose to only show you these results today. And as a brief conclusion, so what we tried out was more complex architectures than FNNs. So we also tried CNNs and different types of data sampling techniques. And but what we saw also in other use cases was that embeddings capture local residue environment. So we don't need a convolutional layer, which you might be able to introduce some more context. So you can take different window sizes and add some more input about the residues around your residue of interest, but this doesn't actually improve performance. So lean architectures with an FNN with just one hidden layer already provide state-of-the-art performance, which is great because it also shows that the language model really captures a lot of information. But as for many use cases, we do need additional experimental data to actually train larger scale models. And yeah, just to deepen the understanding of these molecular interactions. if you do want to look at our work, there's a preprint GitHub repository. You can always reach out to me if you want to. And I would briefly like to thank Maria, who really advised the work a lot, and then the entire Ross Lab, the Elixir seminar organizers. Thank you so much for inviting us. And then, of course, the group at Padua, who's very heavily involved in this research, and all other expert curators and contributors to these databases. And thank you all for listening. I tried to keep it brief, and I would like to answer any questions that you have, and Michael would surely too. So thank you.
Thank you very much, Céline. So now questions by the audience are permitted. So please go on, raise your hand if you would like to, pose a question orally. Because, we had several questions on the chat to which I think Michael responded. I don't know if you had any questions to you, but I think it would be nice, to have questions from the audience. Okay, I have a question for you, Céline. Did you use any other information than, the annotated binding site? Did you use also, secondary structure information or local structure, because there is a lot of correspondence, between I would say, the population of semi-structured regions and the binding regions. In other words, binding regions are usually those that, become structured upon binding and hence already sample, a local structure. So did you think of adding this information somehow?
You mean as input or to predict it?
Yes.
Yeah. No, so we tried to keep it as lean as possible, but this would certainly be something we would be interested in looking at. So if we can also add other information such as conservation, for example.
Yeah, that's of course, conservation, but also, the trend, the propensity of adopting local structure, and that you can get also from the IP community. I think Wim Franken has developed a method where they use NMR data to predict the dynamic behavior of these regions, and to see in the binding region there is a correlation, but of course it's not clear cut. But I think these are information that you could probably use in some way or another at some point.
Yeah, thank you for the suggestion.
That's really it's tough. It's really tough, especially since that these ordered regions tend to be multifunctional and multivalent and so on. So
yeah. So we've definitely run into, I wouldn't call it a problem because it's a very interesting challenge, but just to get ground truth labels that are actually predictable or generalizable to other settings, I think that's certainly something.
Do you know about the work that is being done at the EPFL at Bruno Correa's group?
Yeah.
To predict binding residues in structured regions, and they have a really nice performance for that.
Yeah, he's actually also involved with VantAI, so I was lucky enough to talk to him before.
Okay, great. Other questions? Questions to Michael? If not, I also had a question, Michael. It seems to me, you're looking at some point we're talking about Euclidean distances in the embedding space.
Yeah.
Embedding space is really complex. I mean, Is Euclidean distance, you know, a valid distance?
Good point. We tested this also couple of times in the past, and we also compared different distance metrics, including cosine similarity or cosine distance. Turned out that it did not make too much of a difference. And after all, conceptually, Euclidean distance is super simple, very intuitive, so we just stick to it. But other than that, yes, absolutely. I keep being surprised. 1024 dimensions is something where actually this curse of dimensionality should hit you badly, but
and in many generative models, you know, they're using, you know, this so-called embedding, you know, the latent space. It's some representation of latent space. Try to navigate, you know, between structures, between sequences, and you know, it's a big deal, you know, to find out the right way of navigating, you know, where it takes you in real space.
yeah. Absolutely, and this is also why I had actually hoped that we can provide a more explicit, stronger, so similar like protein and PNN, when you go for inverse folding and try to find different amino acid sequences that still fit to the same structure, that you can actually use this structure input as a conditioning tag to guide the generation aspect, so that you only generate protein sequences that really fold into the structure that you gave at the beginning, where you hope that all the sequences you generate are then also performing still the same function due to having the same structure. That being said, after all, you're perfectly right. It will also be interesting to see what happens if you just poke the embedding space during generation a bit and see how the model reacts actually, how it's different. We didn't do this
I was
yet.
fascinated by this because, you know, when intuitively you think that, you know, it's a reduced dimension and it allows you to do much more than you could do in any other way.
Yeah.
So it could be really, you know, fruitful.
Absolutely, yeah. But I guess this is then also more going in the direction of the diffusion or flow matching methods. Coming from Hannes Stark's reading group yesterday, I mean, there is this D-flow where you essentially have more or less this, that you see how you guide the generation aspect thereof.
Okay. Now I had another question, because I don't know language models very well. You know, the tokens you use are usually always single amino acids?
Yes. So this is what turned out to be extremely useful for us. I mean, after all, many of our prediction tasks later on are then also defined on the level of single tokens or amino acids. So we could also cut the input sequence length by considering 3 mers, for example, or 5 mers. And this is also what people did for DNA sequences, because they are much longer. But for us, most of the protein sequences are actually way below 1,000 amino acids, or on average, even below 512 amino acids. So we've figured that it's better for a practical downstream application perspective to keep this nice alignment. Exactly.
Because that allows you to do the alignments. Otherwise, it becomes much more complicated.
Exactly. Yeah.
Fantastic. Very interesting.